Welcome back, friends. In this video tutorial, we'll be talking about the developmental biology basics. As you see, this video is going to be about those students who don't know about the developmental biology very well and exactly about the basics of developmental biology and what do we talk about developmental biology so much and why developmental biology is an important topic to study. Now, for my case, I know that developmental biology is one of the most important and interesting subject of all, because in this case, it teaches us how a simple cell containing the same type of genetic content like the somatic cell slowly turn itself and produce a new offspring, completely new, healthy, functioning human being or whatever organism it ultimately produces. So let's talk about it in much more detail. Say, in the developmental biology, we must talk about what is development. The definition of development I can give you is something like that: a series of progressive changes that generates the structure of the organism throughout the lifespan and the life cycle. That is called as a development. So a series of changes mostly in the form of cellular changes, genetic interactions with each other, cell-cell interactions with each other, give rise to beautiful structures. And see, the beauty of development lies in this simple form, if I tell you, because the beginning of any development process, there are two single cell containing n number of nucleotides, n number of chromosomes in it, and those two cells, one of them is from the mother origin, one of them is from our father. The father origin is containing a flagella, obviously it's called the sperm. The mother origin is called the egg. So those two cells, when they fuse together during an event called fertilization, after that event, those two n number chromosome fuse with each other, mixed with each other, and now what they have is 2n number chromosome containing larger cell. Though that cell cytoplasmic content is derived from the egg, but still the nucleus of both mother as well as father are fused and they form a giant nucleus, 2n number of chromosome containing nucleus. And this is the only cell, believe it or not, this is the cell everyone should have at their earlier life stage, and that cell will finally differentiate. They will divide, differentiate, and ultimately give us the fully functioning cell or organism. Say all of us started our journey from that 2n number of chromosome containing cell, from that cell which we call as a zygote. Every one of us started our journey as a zygote, and then cell division take place, cell-cell interaction take place, tremendous amount of genetic interactions take place, and finally we are developed. So see, whenever we see a child in front of our eyes and we see that child going bigger and bigger over time, growing bigger and becoming an adult, it's a kind of treat to our eyes, right? For the parents, when they see their child growing from very small to the adulthood, it's a huge, this feels very strange as well as very pleasureful at the same time. But can you imagine, we see a child, that child has the same things, 2 hands, it has the head, it has everything, just everything got bigger and bigger over the time, they grow over the time. But can you imagine, if you start with one cell, it's a 2n number of chromosome containing cell, very small cell. Can you imagine that small cell and slowly start dividing and finally provide us that child? One cell differentiated into an organism. This is beauty, pure beauty of nature. So see, this is the 2n number of cells. There are some important concepts of developmental biology. First concept is concept of cell division. Cell division. So that cell, that single cell, 2n number chromosome containing cell or zygote, start dividing and they will give us huge number of cells all packed with each other. So this is called as a blastula. The phase that is called blastula. And blastula means a lot of cells. They are all originated from the zygote, divided and they are packed together like a mass of cells. Once that thing is done, then those cells slowly start to arrange themselves as a surrounding a small hole or creating a small pore in the middle. That small pore that is created is called blastocele, and those cells that are surrounding the blastoceles are called blastomere, and the phase is called blastula. From the morula, when the cells start to organize itself, we call it a blastula. And then that blastula undergoes a very important differentiation phase, a cellular differentiation, where each of those cells till blastula, each of the cell containing same number of chromosomes, same genes, everything is the same, and the expression of genes are also the same. But after the blastula, it enters a process called gastrulation, in the gastrula phase. That gastrulation is a process which determines that which cell is going to produce which type of organ in the body. This is the time when cells are destined, they are differentiated, they are provided with their information that this cell is going to convert into the liver cell and this cell is going to convert into the nerve cell. So this is the differentiation that occurs during the gastrulation phase. Once that destination code is provided to each of the cells and they provide the lineage of different cells, then after that we call it the morphogenesis, where morphogenesis is a process of slowly accumulating changes over the time to ultimately develop into the designated organ structure. But for the morphogenesis, a cell should have a proper instruction to grow, and that instruction they got is from the gastrulation, right? So see here, after the gastrulation, this is the gastrula phase. After this gastrulation, organogenesis takes place, morphogenesis takes place, and finally you will see the complete organism formed. In this case, it is a starfish. So this process of development or slowly occurring changes over time includes the growth. I've told you, this is the first thing, the cell division and growth. Second thing is the differentiation or gastrulation. This is the differentiation phase or gastrulation process. And third is the morphogenesis phase, which converts those designated cells finally into the organs and to make a mature organism. Another thing you should know about the developmental biology is that the determination precedes in differentiation. Differentiation means those cells that we see, they are differentiated. That means they are designated to produce a specific type of cell, right? So the determination is very important, that which cell is going to convert into what type of organ, right? So that is the determination. Once the determination is done, then only the differentiation can happen. So see here, determination is a commitment of a cell to a particular fate that occurs by the activation of specific genes. So see, once a cell is determined to produce a cell, let's say this is a cell that is present in the blastula somewhere, it is determined to produce liver cells, then a specific type of gene interaction will take place in that cell to convert into a liver cell. Let's say there is another cell which is of the same lineage previously, every cell was same in the blastula phase. After that, this cell number 1, 2, 3, let's say, cell number 3, this cell determines itself to produce a neuron or a nerve cell. So the chemical signaling that will go on and the gene interaction that will go on inside that cell will be different than the interaction that is going on in cell number 2 and cell number 1. Because this cell wants to produce a neuron, this cell wants to produce a liver. So what kind of organ or what kind of cell they want to produce when they are matured, it depends on the determination process. So they are predetermined to produce a specific type of cell only, and they will produce that type of cell only there. So see, and this is affected by the cytoplasmic segregation as well as the induction. And differentiation is the result of the determination. That's what we can tell every time. So see here, the process of differentiation and the determination that a cell is going to be, this A cell is going to be a nerve cell, B cell is going to be a liver cell. So that determines the content, the cytoplasmic segregation and the way the cytoplasmic segregation occurs. This is one thing that plays a role, but majorly the role here is played by the gene interactions inside the cell. The gene, how they express inside the cell. Because remember, all the cells contain same genetic material, same genes. It only the expression that varies the cell. right? The expression of certain genes in neuron cells are more than the expression of other genes. So there are the same genes that are present in a liver cell as well as in a pancreatic cells as well as in the neurons. But in the pancreatic cells, there are some genes that are responsible for secreting insulin is important, so they are expressing that gene. But in neurons, they won't express those genes which is producing insulin, right? So this is the idea. They are specializing it. Everyone is treated with it. For example, when you are studying something for a bachelor's level, let's say bachelor level bioscience, everyone was given with everything, every biology concepts and everything you study in details. But after that, in MSc, you are specialized in a specific topic. Though you know all the rest of it, you have studied rest of it, but some people will be given some special topic to read more and only do the job for that part. So that is how simply things going on, specialization is going on in your body. So say here, this is a cytoplasmic segregation. So this is there are two different methods of cytoplasmic segregation is possible. This is one method where we segregate the cytoplasm horizontally, and this is another method while we are doing it vertically. So see, if we do the horizontal segregation, what will result? It will result in two different type of cell. For example, these are the two different poles of the blastula, because when the cells start to divide, it start to originate two different spheres of cell. If we conclude, the two different hemispheres are called, one as called a vegetal pole, another is called as an animal pole. Usually this one is an animal pole, this one is a vegetal pole, right? So here if you divide them, if we segregate animal pole from vegetal pole, what will happen here is that animal pole contains less yolk content. That's for true for most of the embryos. Vegetal pole is the region which contains more of yolk content. So here the animal pole will ultimately lead to the generation of this embryonic cell and to produce certain cilia. It will remain as an embryonic cell, and it will not differentiate further, and it will not produce any organ. It will not produce any individual. But on the other hand, the other four cells here from the vegetal pole, if you look at there, it contains yolk and everything properly distributed. So it will produce larva, but the larva will be abnormal. It will not be functional. Okay, so this is not a good idea to separate animal pole from vegetal pole. But say the second division, that is a vertical division. Now if we segregate the cytoplasm vertically and segregate them into two different parts in such a way that both of those parts receive some amount of animal pole, some amount of vegetal pole from both the sides. See, both of them receive some amount of animal as well as vegetal pole. Then they differentiate and they will produce normal larva, normal fully functioning healthy larva, but they will be smaller in shape and structure because as they lost the content of the cytoplasm. So this is the thing. So it is giving us an important insight that the division and the segregation of cytoplasm is very important. You cannot segregate them horizontally. The vertical segregation is better because every cell, every embryo require its animal pole as well as vegetal pole for the proper development. The third important concept I want to talk about is the induction. See, we talked about differentiation. We talked about the whole process and how the development works. So we talked about the development overview. We talked about the differentiation overview, and differentiation depends on segregation and all those stuff. Now the third important concept is the induction. Induction is a process in which a substance or tissue influence the fate of a group of adjacent cells, right? So we've seen that differentiation is important. For the differentiation, determination is very important. Once a cell is determined to produce a specific type of cell, then that cell will grow and produce that cell lineage. But induction is occurred after the differentiation is done. When a cell knows what kind of cell it has to produce and it produces that type of cell, then that cell influence, that differentiated cell can influence nearby cells, adjacent cells to be modified into a different tissue. For example, a common example for that is this forebrain influences the formation of lens and cornea. Okay, So why? Let's say here this is the development of a frog, and if you see, this is a frog brain region. This is during the development only, it's not still not formed. So the part which is differentiated to produce the brain, which is differentiated to produce the part of the mouth, that is the eye, brain and all this stuff. So see, this is what we call as a frog head. Excuse me, we call it a frog head. And this frog head, it is having a particular section called developing forebrain. This is the region which will develop into a forebrain of that frog. And see, it is surrounded by a surface tissue, simple small layer, thinner layer of a surface tissue. And these are destined. The surface tissue that is present is not destined to produce any kind of structure yet. But the structure here, we see the head structure is destined to form a forebrain, that means it's destined to form a brain and front face of the brain. So if we zoom in here, this particular structure of forebrain, it slowly start to push that surrounding tissue or surface tissue, and then slowly start to engulf. Actually, it start to arrange itself in such a way, not engulf, but see here, this is the forebrain, start to form a structure like a cup here. As it is trying to make a structure like this cup, and it is also trying to make interaction with the surface tissue. So surface tissue also have a structure according to their presence of this forebrain, and forebrain forms that kind of cup structure which we call as an optic cup. See, when it start to form that optic cup, that cup influences this nearby surface tissue. It influences the nearby surface tissue. This is the surface tissue, and the surface tissue gets influenced due to the formation of this optic cup. The surface tissue cells slowly start to arrange itself, so start to grow, and microtubules that are present, actin filaments that are present in those surface tissues slowly start to grow, and then those tissues start to modify, those cells start to modify, and then start to bend itself in this way, and then we call them as a lens placoed tissue. And then finally those tissue will start to form what we call a round like structure. So they will be fused with each other and to form a round structure like that, which we called as a lens, right? So see, the optic cup that is formed, which is an extension of the forebrain of frog head, helps to form this lens, and then after that cornea will also form. But actually the idea here is that one particular tissue which is differentiated, in this case the forehead tissue, which has that forebrain region, influences the surface tissue to produce itself into a lens. This is called the induction. Induction from the forebrain towards the surface tissue to form the lens. So see here, another example is provided. So here prism, so this is the process where you see this is the gastrulation phase, where we have the different tissues, layers originated, because during the gastrulation phase different tissue layers start to originate, ectoderm, mesoderm as well as endoderm. Ectoderm is a part which surrounds the cell, that is making us the skin, that helps to make the epidermis, it helps to make the nerve system and everything. Endoderm start to help us to make the digestive system as well as the respiratory system. Mesoderm helps to produce, blood vessel as well as the other systems over there. So see here, in this case blastoceles formed, and the idea here is that this determination is very important. So once a tissue gets determined, once a type of cell during the gastrulation is destined to produce a specific type of cell lineage, and if you take that cell out and place it during any other blastulation phase of the same type, it will develop into that tissue because it is already getting the code. But if before the embryo hit the gastrulation phase, during the blastula phase, if we take some cell out there which is not destined to produce that specific cell type, and then we put it somewhere in the embryo, It will not develop into that organ anymore. So see here, in the blastocyst, we take that part, and if you put it here, the neural tube, the neural tissue, if you put it here, it will produce two different individuals like that. Once we talk about these basic concepts, these are up till this part, whichever we talk about is the basic part or basic concepts about the developmental biology. The basic concept means the developmental biology works as a teamwork where cells interact with cells, where genes interact with genes, and cellular interaction plays a vital role to produce and mature organism. But over the time, another thing that is important, which is the differentiation, where a cell is destined to produce a particular type of cell only, because before that every cell are of same type and they are not differentiated. That means they are unique to everyone. They can produce any cells. They are totipotent in nature because they can produce totally different type of cells from that single cell. But as the time goes by, specific cell start to acquire some specificity, and cells, some destined to produce liver cells, some destined to produce digestive system cells, some destined to produce neural cells, some destined to produce blood cells. So that's how the new lineage of cells start to originate, and that requires a determination phase, and determination works earlier. Once the cell is determined what kind of tissue it's going to produce, then the differentiation takes place. And the third important concept is the induction, where a one type of differentiated cell or tissue influence the nearby cell to produce a different organ, right? In this case, you see the example of the forebrain of frog helps to differentiate and produce the lens for the frog, right? So now we'll be talking about the process, the differential sequential processes for development. And the first thing is the fertilization. The fertilization process is the most important because as this process will ultimately lead to the generation of those zygote, that first cell, diploid cell, 2n number of chromosome containing cell. Once the zygote is produced, then that zygote will slowly divide and divide to produce blastula, then gastrula, then the other stuff. So see, the first stage of fertilization. The fertilization event, the basics are very easy, where we have two different cells. We have a larger cell, which we call as an egg usually, egg, and which is produced by the female, right? And then what we have? Then we have sperm cell, which contains a flagella. So both of them, egg as well as sperm, contains n number of chromosomes in the nucleus. Sperm is the cell which migrates, which moves, and finally go and interact with the egg cell and donates its nucleus inside the egg cell. Egg remains as it is. Egg holds on to the nucleus coming from the sperm. The nucleus coming from the sperm is called as a sperm pronucleus, and the nucleus that is present in egg is called egg pronucleus. So after the sperm donates the nucleus into the egg, then both of the nucleus will fuse. The n number of nuclear content, chromosome content, will fuse to produce a 2n number of content, and the egg cell slowly becomes slightly larger, and then it produces what we call as a zygote. Whenever the 2n number of chromosome is made after the fertilization, we call it a zygote, and then the zygote will undergo cell division and differentiation to finally make the embryo. So here the first step is a contact between the sperm and the egg cell, and that we can see here in this picture. This is the egg cell. You see a lot of things, lot of receptors are out there for the reception of the sperm, because sperm has a specific signaling molecule and the egg surface has specific receptor for that. Then the second is the contact between the sperm and egg's plasma membrane. And when there is a contact between sperm and egg's plasma membrane, then the nucleus slowly start to insert itself into the egg, inside the egg cytosol. And then remember, sperm does not donate any cytosol. Whatever cytosol that is present in the zygote came from the egg. Sperm only donate its nucleus. So this is the process of fertilization. See, at the very beginning, binding of sperm to a specific region, which we call it different cases. In case of mammals, we call it a zona pellucida. So see, this is the process. And sperm has a specific structure. If you look at the sperm structure, it will look something like this, for example, and kind of. And here we have specific things, because what I've drawn this structure, because this is the nucleus and this is the specific regions. So if I designate this different regions, this is the flagella. Remember, with the help of flagella, sperm can swim. Sperm can sense the presence of chemical contents as well as toxic materials. So the movement of sperm based on the chemotaxis. And this part is called the collar region of the sperm. The collar region of the sperm contains mitochondria, because sperm needs energy. A lot of energy is required for the movement of sperm and finally reach the egg, right? So here the sperm move by rotating its flagella, and by finding the chemical gradient it is observing. But the energy is derived by the ATP hydrolysis, and the ATP is provided by the mitochondria. It's produced in mitochondria. And this is the body of sperm, which contains a nucleus, sperm pronucleus, and at the top, in front of that, it has a structure called acrosome. Acrosome is a content which is filled with vesicles containing enzymes, digestive enzymes. Those enzymes can digest the vitelline membrane of an egg, because egg has a membrane, multi-layer content. Where is a cell membrane? First a cell membrane, then a vitelline membrane outside, then it has a jelly-like coat outside. So jelly-like coat is for the prevention. That can be easily destroyed when the sperm take entry. But the second important thing is to break the vitelline membrane prior to the development and release of the nucleus inside the egg cytosol. And for doing that, it needs to break it down. And for breaking it down, it requires those enzymes that are present in the acrosome, right? So acrosome contains digestive enzymes. So see, binding of the sperm to the zona pellucida occurs. After that, the acrosome reaction takes place. Acrosome reaction means acrosome slowly secretes some of the materials outside, and it is going to degrade all of these other debris and all those vitelline membranes and all this stuff from here. As a result of that, the sperm is in direct contact, and it can fuse its membrane with the membrane of egg, and then the cytosol of sperm and egg can mix. Is in contact, and in that condition only the egg contents are provided. The egg content, the sperm content, that is the sperm nucleus, sperm pronucleus, is provided inside the cytosol of the egg. So these are the sequential images that are given here. Remember, this thing never happens in multiple chromosomes, multiple eggs. One sperm only can fertilize one egg, and that is a process very tightly regulated, because an egg cell does not want more than one sperm to fertilize it, right? Those sperms are produced in huge quantity. Most of the sperms lost in the battle, only one sperm win, and that sperm will ultimately fuse its nucleus with the nucleus of the egg to produce a zygote. Sometimes two sperms can fertilize an egg, but that's a different case, but very rare case. Most of the time it is not allowed, and we call this blockage as a polyspermy. So see, this is a process, an example in sea urchin, that we can see that the process of interaction between sperm as well as egg is due to the interaction between ligand receptor binding, where the receptor that is present on the surface of egg is called the binding receptor, and the chemical molecules that will bind, that will help the sperm to bind with the membrane of egg is called the binding molecule. So binding molecule is on the top of the sperm cell, and binding receptor present on the surface of egg. With the help of this binding molecule and receptor, they interact, and the sperm is fused with the egg. Now this region, which we call a vitelline layer, is very equivalent to the layer of zona pellucida that we see in case of mammals, because in case of mammals we don't see that vitelline layer, instead we see that zona pellucida. But in either way, there are interactive molecules, there are ligand receptor binding that helps to stabilize the sperm in direct contact with the egg. Once the sperm is stabilized onto the surface of egg, then sperm start to release this acrosomal content, which we call as acrosomal reaction, and then whether it is a zona pellucida or a vitelline layer, sperm take entry. The nucleus of sperm can take entry inside the egg. This is a process again denoted for the sea urchin. So see, this is the vitelline envelope in between jelly and inside the egg membrane. So with the help of acrosome, it will bind. An acrosome process means, when the sperm is in close contact with the egg outer layer or egg jelly like layer, whatever it is, in that case, you see there are some actin filaments arranged there at the top of the head of the sperm, which start to arrange itself and push these acrosomic vesicles forward. This is called the acrosomal process. So it becomes very sharping, and it becomes very pointy at the top, where all those vesicles start to be very close enough at the beginning, and then when it is in contact with the membrane, then these acrosomal materials are released. So all the membranes that is present on the surface of egg slowly start to degrade, and the nucleus from the sperm can take entry to the egg. Actually, sperm head is very small. It is, see, the cytoplasm to nucleus ratio is not very good because the nucleus is big for the sperm compared with the cytosolic content. This is another case where you see the sperm eggshell content triggers the acrosome reaction, and this is an example for the mammals, right? Because in mammals we don't have that binding and binding receptor. Instead, we have a ZP3 receptor or ZP3 receptor, whatever you can say, ZP3 receptor. Sperms have those ZP3 receptors, and here we are having those molecules in the zona pellucida called ZP3, ZP2 and ZP1. These are the carbohydrate molecules, the glycan molecules that are present onto the surface of egg cell for mammals. ZP3, ZP2, ZPA, they interact with themselves to form a chain-like substances. But here, for the binding with the ZP3 usually presents on the outer side. So the sperm contains ZP3 receptor onto the surface. With the help of this ZP3 receptor that is present in the sperm surface, sperm interacts with the egg ZP3 region, and then sperm can fuse its membrane with the egg membrane, and the nucleus of the sperm can be inserted inside. Okay. So once the sperm is in contact with the egg here with the ZP3 receptor, the ZP3 receptors slowly start to bind, and all the other ZP2 receptors slowly start to degrade due to the secretion of acrosomal enzymes. But remember, ZP3 does not degrade very much, it remains as it is. ZP2 and ZP1 contents are degraded due to the acrosomal enzymes, as well as some region of the ZP3 content, including the membrane of which we call the zona pellucida, which is the membrane for the egg, slowly start to degrade so that the nucleus from sperm can take entry. This is in case of mouse, where you see again acrosome is present. So same case every time in contact with zona pellucida, these acrosomal contents are mediated. They slowly start to degrade zona pellucida, and then the sperm heads slowly start to move inside. And whenever they are in contact with egg membrane and the contents of acrosome slowly start to degrade the egg membrane, then the nucleus, the pronucleus, is driven inside. It is donated inside the egg cell cytosol. So whenever we are talking about fertilization, the thing we should not escape is polyspermy. There is always a chance of polyspermy. Why? There is one egg, there are multiple sperms, so there is a chance that more than one sperm can fertilize an egg. So that is called a polyspermy. That is multiple. Poly means many. Sperm can fertilize one egg, and that is not tolerable. That is not what development works. Okay, so we have to prevent multiple sperm to fertilize one egg, because it's found that this type of thing, that multi-sperm fertilizing an egg, leads to malfunctioning individual, and ultimately it is lethal because it will ultimately kill the embryo. It will not be differentiated into the organism. So it is not wanted. If a sperm only fertilize one sperm fertilize one egg, it will generate into a normal individual. So it's always very important to block polyspermy, and to block polyspermy there are several different approaches, and the fertilization process is extensively studied in sea urchin, because in this whole process of developmental biology there are different phases like fertilization, blastulation, gastrulation, and organization with different phases, morphogenesis. Each of those phases are studied in a different specific type of cells, okay, specific type of organisms, which we take as a say as our organism, as our what we can call it, default organism, say like drosophila, like sea urchin, like C. elegans and all this. So these are the organisms, like the extensively study of fertilization studied in sea urchin, while the morphogenesis is better studied using drosophila. So these are the things. A neural development studied better using C. elegans. So these are the model organisms that we take for the study. So here, blocking of polyspermy for sea urchin study, we see there are two different ways a polyspermy can be blocked. First is called the fast block or transient block. Transient block means the block will stay for a while only, which prevents the sperm fusion to the egg. The second block is called the slow block or permanent block, that is the removal of any other bound sperm that is present to the embryo, that is present in the surface of egg. So here you see the first block, that is a fast block. Say this occurs in 1 to 3 seconds after the fertilization and can last up to 1 minute, and this is nothing but a huge sodium influx to the cell. This is the egg cell, and upon the fertilization event, say one sperm properly attached to the egg surface, it delivers its nucleus inside. Once the sperm pronucleus is inside the egg, we know that fertilization is complete. One sperm has done that fertilization properly, so we don't want any other fertilization event, any other sperm to, we want to prevent we want to prevent the fertilization of that egg by any other sperm. For that reason, the fast block works when a sodium ion, a huge content of sodium ion was taken inside. So it's called the sodium influx. Influx means the sodium concentration inside the egg will start to rise, and they take a lot of sodium ions from outside. You know, in sea urchin, as they present in the oceanic environment, sodium is very common outside in the environment. So it will take the sodium ion, drag it inside. Sodium influx blocks the sperm to fuse with the egg anymore, so no sperm can fuse. Now there are say multiple sperm fuse somehow earlier, and one sperm donates its nucleus. So any other sperm, if they are bound with this egg, in that case we need to release those sperm out, break them out to the environment. So that is called the slow block. It occurs 20 to 60 seconds duration time, and this type of block is permanent. After the slow block is established, then no any other sperm can interact with the egg and can fertilize the egg. So for the slow block, the idea is the internal cell signaling process inside the egg, where you see the signaling process goes on like the phospholipase C. Phospholipase C is a membrane bound signaling molecules that is present, and upon activation it is broken down into two components. One is diacylglycerol or DAG, another one is inositol triphosphate, ISP. That ISP released outside, and diacylglycerol remain attached to the membrane. So this ISP finally interact and with the endoplasmic reticulum that are present in the egg cell, because you know endoplasmic reticulum is present and endoplasmic reticulum has certain type of receptors onto the surface of it. So while that INSP3, the inositol triphosphate 3 bound with those receptors, those are calcium receptors, and they are open, they are calcium channels, right? channels through which calcium can pass in and out of the endoplasmic reticulum. Once that binding of INSP occurs there, that channel opens and calcium slowly start to come up or come out from the ER to the cytosol. So as lot of calcium start to insert itself in the cytosol from ER, huge calcium concentrations start to build there, and as a result of that, the slow block occurs, right? And what it does actually, these huge calcium concentrations also help certain granules that are present in the egg cell near and close to the cell membrane. We call them cortical granules. Those cortical granules contain certain type of chemical factors and also some enzymes which again blocks the polyspermy, and also which will interact with the cell membrane and contain and coat the membrane in a hard form, which will be the egg membrane, the hard egg membrane. So that thing will be produced by this cortical granule fusion, and that is mediated also by the huge calcium concentration in the cytosol of the egg. And also the diacylglycerol that remain on the membrane, which activates another protein or protein kinase C. That protein kinase C helps in exporting protons from the cytosol of the egg to outside, and as a result it increases the pH inside, and increased pH inside helps in the protein synthesis, and that protein synthesis will lead to the signaling that fertilization occurred properly, polyspermy is properly prevented, and after that cell division should work. A lot of cell division will be done and morula stage will be produced. So the question is, how do we know that the polyspermy actually prevented? The idea that we take sodium ion and also increasing content of calcium ion, the exact idea I forgot to mention there, that sodium influx blocks the polyspermy, calcium influx also blocks the polyspermy. The idea behind it is that sodium ion as well as calcium ion, they are positively charged ions. They slowly change the membrane potential, because membrane potential plays a vital role for the fertilization event. For the fertilization event to occur properly, there should be a standard membrane potential present throughout the membrane of the egg for the sperm to fuse with the egg. But now, due to the sodium influx as well as the increased content of calcium ion inside the cell, that membrane potential is disrupted. The membrane voltage slowly changes. As a result of that, that changed voltage will prevent the sperm to fuse with the egg and deliver its nucleus. So see here, normally there is a resting potential of -70 millivolt that is present throughout the cell membrane of the egg. But once the sperm is added, once one sperm is added and fertilization is done by one sperm, then rapidly after that sodium intake as well as calcium uptake there, as a result of those features, that resting potential increases up to 0 to 50 millivolt. So as a result of that, at this lower voltage content, where is the resting potential of -70 millivolt, sperm can fuse with the egg. But once the membrane potential rises to +50, +20 to +50, sperm can't fuse with the egg anymore, so the polyspermy is prevented. So see here, for the slow block also and the mature block, the slow block means the permanent block. For this block, cortical granules plays a vital role, which is poised for release. So see, there are kind of 15,000 cortical granules there are present in the CR chain, and that contains the enzyme that clip the X binding receptor and any attached sperm. So remember, in case of CR chain, in the surface of the egg there are receptors called binding receptors, and there are sperms on the top of the sperms, on the head of the sperms there are molecules called binding. With the help of that binding, the sperm will interact with the binding receptor with the egg and they will fuse. But here, due to the fusion of the cortical granules, the cortical granule fusion releases certain enzyme which will block those surface binding receptors. So blocking all the binding receptors will not allow any other sperm to bind, and all the sperm will fall off. Okay, as well as this same thing changes the vitelline envelope or alter the vitelline envelope. For example, in case of mammals, it is a zona pellucida alteration. That thing is also done. It's also helped to stabilize the membrane and create a hard coat around the membrane, because now the cell will need a lot of cell division. No fertilization is required anymore. See, in case of the mammals, we see again the sperm have ZP3 receptors, and we see the process that ZP3 receptor in the zona pellucida. There are 3 receptors, ZP3, ZP2 and ZP1. Upon interaction with the sperm, ZP2 and 1 are degraded, ZP3 however remains, and then it is dragged inside. The same things there, ZP3 remains there, and then ZP which is present here is a modified ZP3, and this modified ZP3 is required for preventing any further interaction of a sperm with the egg cell surface. So see, this is a realistic image, the real image that we see using the fluorescent dye. A wave of increased calcium can be visualized inside the egg cell in case of sperm entry. So see, this is a condition where the sperm entry takes place, and after the sperm entry is done, then slowly from the endoplasmic reticulum, you know, where the endoplasmic reticulum is present, we see a lot of calcium slowly start to come out. So see, this is the region where it slowly start to come out, and slowly as it is coming out, it is transferring itself and distributing itself throughout the cell for the cortical granules to fuse properly. Okay. So we've talked about the fertilization properly. So once the fertilization is done, that means we know there is a sperm, there is an egg, fertilize itself. So now we produce a zygote. A zygote means it contains 2 in number of chromosomes. Now that zygote is ready for growth. That means division. Rapid cell division should be done for the zygote to produce what we call as the morula stage. Morula is nothing but a mass of cells. They are added one after another, just like a kind of tumor cells. So see here the process to develop morula, including this process called cleavage and farrowing. Cleavage means rapid cell division occurring one after another, but the cells are kept together by some receptors, cell surface receptors like CAMs as well as cadherin receptors. So those cadherin molecules and CAM receptors help those cells to be interacted, to be in close proximity, because we don't want those cells to go away. We want those cells to be together, close together, because after the morulation, it will undergo blastulation, which will be the proper organization. So see, in the cleavage process, it's nothing but rapid cell division. It's a rapid cell division. So see, at the beginning, this is the cell, slowly start to divide. The zygote actually, 2 in nucleus, it divides. It can divide in 2 different ways. The cell can divide vertically or it can divide horizontally. Now this cleavage is varying extensively from different type of organisms. The cleavage pattern is different for a chick embryo, it's different from a human embryo, it's also different for the sea urchin, it's also different in case of frog. So we have different types of cleavage pattern originated, but the actual goal is to produce multiple number of cells very fast. So see here, it is a vertical division, and now it gets 2, then again another vertical division at this angle, and we get 4, and so on. After this division, we have a horizontal division to get 8. So this is the way of rapid division slowly start to work. Now the question is how a cell divides so much fast, because though that cell, once it will produce an adult cell, we'll see that cell division time is not that much fast, but how could a cell divide that fast during the process of morulation and cleavage? The result, the answer for this question is that during that stage of blastula formation and morula formation, actually the cells divide because they divide fast because they have only two different stages. One is the mitotic phase, another one is the S phase or synthesis phase. No other cell cycle phases, because we know normally a cell have many cell cycles phases, mainly one interphase and one mitotic phase. Interphase contains 3 different stages, G1, S and G2, and it took time in the interphase because the mitotic phase occurs really rapid in normal cases also. But G1 phase and G2 phase are the highest time consuming phases of the cell cycle. Now during the process of morulation and cleavage of the embryo, they exclude that G1 and S phase, G1 and G2 phase. They only took S phase as well as the M phase. So S phase means why we need S phase. S phase is a synthetic phase where all the DNA would be duplicated. that is required. We need duplicated DNA to be transferred to the daughter cell. So that is required. DNA duplication occurs at the S phase, and then rapid cell division occurs at the mitotic phase. So only two phases for the cell cycle while it is going on cleavage and farrowing. So see, the molecules cyclin and CDK are required for the cell division, and we see mitotic cyclins are required. So mitotic cyclin, or we can say cyclin M, in this case the example is cyclin B, and CDC2, cell division cycle protein 2, along with the cyclin B. They interact together and form what we call mitosis promoting factor or mitosis promoting complex, whatever, and that complex helps in the mitosis process properly. And then after the process is done, then cyclin, every cycle the process is done, cyclin is degraded using ubiquitin tag, and then again synthesis phase, the cyclins are made as well as the DNA is duplicated, then again the process, see, cyclin is made, DNA duplicated, and then again the process M phase occurs. So this is rapid M phase and S phase, M phase and S phase. No other phases are there, no other growth phases are there, only these two phases. And see, mitosis promoting factor or MPF activates and it turn into. Now this process of rapid division of M phase and S phase, M phase and S phase will come to an end when the cell thought to enter itself into the blastula phase, where everything again turn itself into the normal, and that thing occurs in a mid blastula phase. We call it a mid blastula transition. The process of rapid division occurs throughout the morula, throughout the blastula, and once this embryo determines to go into the gastrulation phase, that means after 20-25 many rounds of cell division, when it's finally decide to go to the gastrulation phase, then from the transition of blastula to gastrula requires the cell to restore its original cell cycle phases, and then it is restored only. So here the process of cell cleavage not only depends on microtubules, because microtubules play a vital role in the segregation of chromatids, segregation of chromosomes, and transferring them to the daughter cell. But not only the microtubules are important, but also microfilaments are playing a vital role, because see, normal cell division occurs slowly, different phases, they segregate the chromosomes, then slowly the cytokinesis take place. But in case of these developmental phases, rapid cell division is occurring, genomes are separated very fast, and the cytokinesis, that is the division of cytosol, also works simultaneously with the separation of chromosomes. So both these things, separation of chromosome as well as cytokinesis, works rapidly, same together, same time, right? So for this thing to occur at the same time, they need these microfilaments, which we call as a contractile ring. So these microfilaments or contractile rings are present as the cleavage portions of the farrow. So this is the farrow region, these are the farrow region, farrowing sections. So from this farrow regions, those microfilaments slowly start to build, and they are to push the cytosol from both the ends. So as they are pushing the cytosol from both the ends, and then they are going to break down the microtubules in between, and then they are going to separate the chromosomes apart into two different sections, and they produce two separate cells. But remember, as a result of these contractile rings that are present in the middle, during the rapid division, cell won't create any damage, but also these microfilaments will hold those cells very close to each other even after the division, and then the surface receptors of the cell will interact to hold the cells together. Because what do we want? We want a stack of cells like this. We don't want cells separated in a space like that. Okay. So if you see during the division, this is the 3D image, you see the stress fold or contractile C. This contractile ring made with microfilaments, the job of this is to simply create a stress and close the door slowly, so that microtubules got broken and the cytoplasms are apart. Just take a balloon, water filled balloon, and take a rope. Try to put the rope on the middle and slowly start to put a stress. You will see two lobes start to form at two different sides, and all the water contents will be they are broken into two different parts, and you'll be able to put a tie there or not there. This is how this thing works simultaneously. The stress is applied by the contractile ring formed by microfilaments. And this is an image. You will see the actin microfilaments are working properly for making that contractile rings. Okay. Now I've told you earlier that different organisms have different type of cleavage patterns. For example, say different cleavage patterns, an example like isolecital cleavage pattern or centrolacetal cleavage pattern. Isolecital cleavage pattern means the cleavage will be carried out where the cytosol will be segregated in both the way. Whatever way we divide same amount of cytosol in both the daughter cells. While centrolacetal means the cytosol will remain in the center and all the cells that will be formed surrounding that region. The centrolacetal cleavage pattern is only seen in case of drosophila. See, in case of insects, the example is drosophila. In case of it, you can see the yolk that is present, it should be present in the center, and all the cells that will be covering that center, surrounding that center. See, this is the center region, and these are the cells that are covering the center throughout a periphery. While isolecital condition is the yolk is entirely distributed into the new daughter cells, and the example for that, it could be radial, it could be rotational, or it could be say discoidal. So radial, rotational, or discoidal. Radial cleavage means the cleavage will occur either in straight or in horizontal or in vertical direction. See, example is Amphioxus, the frog blastulation process, frog cleavage process. While the rotational type of cleavage, examples are mammals. Rotational cleavage is different. Rotational cleavage means one cleavage will occur vertically, then another one will occur horizontally. So it will rotate the axis of cleavage, because if you're talking about a cleavage, that means you're talking about breaking something, cutting something into two. So you can do it in different ways. In rotational cleavage, they will rotate the way a cell is divided, the way the microtubules are arranged. So there are different types in case of mammals. But in radial, it will be a set type, whether it's a straight or cross, it will be distinct from before. The third kind is a discoidal. Example is fish, reptiles, birds, all of them have a discoidal case. Discoidal means the yolk-rich region will be present and the cleavage will start to occur slowly on top of that. So see here, this is the egg portion there, and its cleavage will start from on the top, and rest of the portion, maximum region of the yolk, it will remain as it is, and only the cells that slowly start to divide from the top, and they will be arranged itself, they will arrange itself in the top layer of that huge zygote that is present on the bottom. So this is called discoidal image, discoidal cleavage, and we see in this case of bird, fish, and reptiles. So now the big question, what controls the pattern of cleavage, right? What is controlling the pattern? There are two different things that is controlling the pattern of the cleavage. First is the amount and distribution of yolk in the egg, where exactly the yolk is present. If a yolk should present in the center of that egg, if the yolk is present distributed center of the yolk egg, in that case the cleavage will occur in the isolecital, not isolecital, it will occur in the centrolacetal process. Like for example, like in this case of insects, centrolacetal. Why? Because the yolk is present in the center. That's why the centrolacetal cleavage takes place. On the other hand, so if you look at here, it will be much more clear. See, this is the complete cleavage for frog and this is for chick. For the frog, say the yolk content for the frog present in the vegetal pole, because these are the two different poles, animal pole, vegetal pole. See, animal and vegetal pole. See, most of the yolk content present in the vegetal pole. As most of the yolk content present in the vegetal pole, the cleavage will start to occur from the animal pole, from the opposite side. From the animal pole, the cleavage begins. See, simple division. You have a say orange, you start cleaving from top, then like this, then like this. See, lots of cell division like that. After that, the division will be 3 division on the vertical way, then 1 horizontal division. This is a pattern that is coded there, that is present there, and it will occur like that. From a fertilized egg to 8 cell stage, it will slowly divide. On the other hand, in case of chick, we call it an incomplete cleavage. where the yolk is present and distributed. See, the yolk is present and distributed throughout the surrounding regions mainly. So for that reason, in case of chick, the division will not take place where the yolk is in rich condition. Remember, every time they don't want to create any barrier in the yolk, because yolk contains all the nutrients for the development and nourishment of the embryo. So they will not cleave the yolk. So yolk will remain like that. So wherever the yolk is present, cleavage will begin from the opposite side. For example, in case of frog, the yolk is present in the vegetal pole. So the cleavage starts from the animal pole, right? Again, see, 3 cleavage from this, and then again the 4th one, the horizontal cleavage also works in the animal pole, not in the vegetal pole. See, in case of chick, as the cleavage is, as the yolk is present in the surface, in the surrounding region, you can see this is the yolk content surrounding the region. So the only way the cleavage could be possible is a region where is yolk less, a less yolk containing region, and which is the middle portion where we have less yolk. So the process of cleavage in case of chick is never a complete cleavage. So complete cleavage means you start cleaving, say a orange. You take an orange and a knife. You just start cutting the orange completely to the from top to the bottom, and it separates like that. This is a complete cleavage. But in case of incomplete cleavage, you start cleaving and you stop. Again rotate, again cleave and stop, but not complete. You are not cleaving it completely to the bottom. So that's what you see. Whatever we see here is the top view. Remember, this is a top view that we are seeing. These are the side views that we have seen. So from the top view, what we see, that as the yolk is distributed in the peripheral region, so we have only one option to cleave in this blastocele, the black region, and we start cleaving it from the top. See, not a full cleavage, or incomplete cleavage, then again a rotation, again a vertical incomplete cleavage like that, then again multiple incomplete cleavages. All the cleavage will occur from the top like that, incomplete cleavage in case of a chicken. Okay, so it depends on the content of yolk that we have, where exactly the yolk is present. See, this is the discoidal cleavage for birds, for reptiles, as well as for fishes. This discoidal cleavage, you'll see the beautiful example. These are all the real images, the 3D images. This is the egg. The top portion of the egg slowly start to rise itself. Remember why? Because the top portion does not contain any yolk. Most of the yolk-rich contents are present in the bottom. So the top is yolk-free, yolkless. So then the division will start place, and division occurs like that. First division, again, then from this angle, so multiple angle division, then it came for this angle, so it will divide itself, and as a result of this discoidal cleavage, where it creates the area of less yolk containing section on the top, while yolk present in the bottom, and whatever division take place in that yolk less region in the top, and as a result of that, we see a huge content, a huge region, a large section of the embryo at the bottom with yolk, and the top, multiple cells, small cells, multiple division cells slowly arise, and they have a stacked structure like that. It will look something like this. You see multiple cells, whatever division take place on the top, and no division at all take place in the bottom. So that's why we call it a discoidal. It looks like a disc, a floating disc, discoidal cleavage, right? So it creates the cellular region up above the dense yolk, and for creating this region on the top, see some uplifting. If you look at this structure, it should be like that, but some section is uplifted, and actually they produce this uplifting using actin filaments to drag some sections of the cell outside and taking some cytosolic content to the top, so that they can create the animal pole there and can start cleavage from there. It's a beautiful example of how cell interact for not cleaving the yolk. It's never possible to cleave the yolk at the first place. It's never done. And the second factor that determines the cleavage is the factors in egg that influence the angle and timing of the mitotic spindle formation. Because you know, for the cleavage, spindle formation plays a vital role. Say, during mitosis, these are the chromosomes that are arranged, and let's say we have the microtubule organizing centers in both the distance regions like that. They hold on to this chromosomes. If this is the way it originates the chromosomes, like this axis, then they will generate and produce two different cells like this, right? Now let's say in the same case, say sorry, say let's say in the same cell we have chromosomes arranged like this. In that case, microtubules will hold on to the chromosome. Sorry, it is a bad drawing, it's not a good drawing. I should draw here. So these are the chromosomes, and microtubule organizing center is always there. So microtubule MTOCs are there, and they are holding on to the chromosomes from this angle, this angle. So previously the axis was straight 90 degree, but now if we see this is, there is an angle, so not 90 degrees. So then the cell will divide and they will produce two cells like this angle. Previously we produced two cells side by side like this, but now we are producing cells by this angle, this much angle, right? So this is important, the assembly of microtubules, in which angle the microtubules are assembled. So the way the angle the microtubules will assemble, they are going to drag the chromosomes using that angle, and they are going to produce the cell side by side by that angle only. So that is another thing. See here. But another thing I must say, the cytoplasmic determinants that are present is also important. Say here, in case of proteostomes, say in this case we have a blastomere, lots of cells are there, and if we take one cell out of the blastomere and if we place it, if we check for the pattern that emerges, it creates the spiral cleavage, and then finally normal individuals are generated. So see, but if we take a small part of it, if we take a small part out, that small part will not be able to generate an individual or embryo or a gastrulation. But where we have a deuterostome, in case of deuterostome, if we take a small blastomere section, if we take a small cell from the blastula and then put it, normal larva develops, and normal, everything develops into normal thing. So it's not only the cell division that is important, but also what cytoplasmic content that are present in the cell, because genes are present and genes are expressing during that time. They are producing certain cells required for the cell division process as well as cell differentiation process. So in case of protostomes, for example mollusks and annelids, we see in this case, if we take a cell from the blastula, cells that are present in the blastula phase, we call them blastomeres. So if we take one blastomere from that and if we plate it, that blastomere will not develop properly and not giving us anything, right? And the development will be arrested and the cell will die. But on the other hand, in case of deuterostome like echinoderms or chordates, if we take one blastomere out and we place it, we culture it, we give every nutrients, that one cell from blastula will ultimately go the cleavage and all the rounds, gastrulation, and finally give us the embryo, an adult individual actually, right? So that is the difference between what kind of individual we are dealing with. Even in case of chordates like us, those small cells, this one cell of the blastula contains the potential to produce a healthy normal human being. So we call them totipotent stem cells. They are the stem cells, and we store those cells now using different processes. Life cell technologies is doing a lot of stuff like that. So we preserve that stem cell so that if there is any problem for our child in future, genetic problem or any other problem, we can take that stem cell out, we can preserve that stem cell while in the time of birth, then we can take that stem cell in future and use that stem cell genomics and cells to treat all the kind of genetic problems for the person. So this is a huge thing, important thing for new experiments are going on this topic. So but this is a way the cytoplasmic determinants also influence the cleavage patterns that are present. For example, you see the radial cleavage, example is in sea urchin, right? Radial cleavage means the cleavage will a simple plane of cleavage, say straightforward line. Say this is the cleavage from the animal pole like this, then again from the animal pole like that. So the cleavage will occur always in the meridional cleavage, means vertical cleavage. This is another name of the vertical cleavage. Equatorial cleavage is another role of horizontal cleavage. So horizontal cleavage, also known as equatorial cleavage, like that, and meridional cleavage means vertical cleavage from the top. So it will cleave like that. Now we can also divide cleavage based on the type of cell division, synchronous cleavage or asynchronous cleavage. In the synchronous cleavage, even cell stage are generated from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32 and so on. Even number of cell stage generates. Whatever cell combination generated are of even number. On the other hand, there are some processes called odd number cell stages, and that is possible in case of asynchronous cleavage, where the cleavage pattern is not synchronous. That means the cleavage pattern is not properly specified. That sometimes cleavage occurs properly, sometimes it does not occur properly. It does not provide and produce a or even number of stage. Odd number of stages are generated in asynchronous cleavage. And in mammalian cleavage, we have seen before that mammalian cleavage is unique. Why? Because it relies on the rotational method of cleavage. What is a rotational cleavage? Rotational cleavage means it changes the plane of cleavage. You know, radial cleavage that we see, the radial cleavage does not change the plane of cleavage. The plane of cleavage remains stagnant. Either it is a meridional or equatorial, or the combination of two. But the rotational cleavage occurs while the cleavage plane change in every time. So at the very beginning, the cleavage plane was this. In the second cleavage, the cleavage plane changes. In the third place, the cleavage plane changes into equatorial. So first one is meridional in this angle, second is also meridional this angle, third is equatorial this angle, fourth is again meridional, fifth is again equatorial. So it shifts all the different planes of cleavage, and this is very rapid. I mean, every time slashing of a cell to produce a multiple cells from one zygotic content, right? So in amphibian, see either, so in amphibian or echidodon, whatever you take, they belongs to the radial cleavage. Radial cleavage means cleavage 1, cleavage plane 1, meridional, second meridional, third meridional, fourth also meridional, radial cleavage. But rotational, first meridional, second equatorial, third meridional, fourth equatorial. So it shifts the plane of the cleavage. Okay guys, until this part we have studied the process of cell division which give rise to morula phase from the zygote. So zygote was a cell with 2n number of chromosomes, and then start dividing and it produces a mass of cell attached one after another, just like a tumor condition, like the tumor cells, and they aggregate together, right, to make a solid mass of cell. Now that solid mass of cell is then modified. See this is the cell, these are the cells that are arranged together, solid mass of cell. Then those cells start to arrange in the periphery, creating a small pore in the middle, which is a hollow, forming a hollow sphere in the middle. This phase is called as a blastula. Remember, blastula. And the cells that are surrounding the periphery, those cells of blastula are termed as blastomere, and those pore inside is the. So if I go here, it is clear. If you see here, it's something like this. See, this is the phase of morula where cell divide one after another and stack itself one upon another. But in blastula, those cells are much more organized in the periphery only. So those cells of blastula are termed as blastomere, right? And the sphere that is formed, the pore that is formed in the middle, the fluid filled cavity or space that is formed is known as blastocele. These are the two terminologies, right? So morula is a ball of cells, blastula is a hollow sphere of cells. Now the question is, when once the blastula is formed, this is the phase where cells are organized. After the blastula is formed, then the cell will undergo the determination, because each cell has a particular fate, right? That cell will produce a type of cell as the embryo will mature into organism. Some cell will destined to produce neuron, some cell will destined to produce the liver cell, some cell will destined to produce the muscle cell. So once that determination is reached, then the important task for generation of the different tissues and cells, and this process is known as differentiation phase or gastrulation phase. Now during this phase, the fate of the cell is already determined. Now the cell will undergo some changes, some physiological changes and the gene expressive changes to ultimately lead to the generation of the destined specific structure of the organism. So during that point, during the point of cell division in morula as well as in blastula, we have seen the cell division to occur rapidly, very fast. But in this case, once they are restored to the formation of gastrula or gastrula phase, where they want to produce a specific organ structure, in that case, those cells should come back to the normal cell cycle or normal mode of cell cycle. Remember, during the morula and blastula phase, in midpoint of the blastula, the cell cycle that they undergo is contain only two different phase. One is M phase or mitotic phase, another one is S phase. So S phase, synthesis of DNA occurs and synthesis of cyclin occurs. Then in M phase, cell division passes by. So they bypasses the growth phases G1 and G2 phase completely during the process of morula as well as the midpoint of blastula. But after the blastula is reached at the middle, so the midpoint of blastula, that's called the mid blastula, when those cells are containing the code for the determination to convert into a specific organ, at that time those cells should convert its cell division cycle again into normal. So this is a mode of normal cell division where we have a G1 phase, larger, S phase of synthesis, a growth 2 or G2 phase, larger time, and then the mitotic phase. So in this case, they should shift back to the normal, and this shifting is called the mid-blastula transition. While the cell is entering the embryo, a cell division entering from blastula to gastrulation. So see, during this process they will involve multiple cyclins, not the only cyclin B that was taken care during the morula and blastula. So here we have cyclin B, along with we have cyclin D, we have cyclin E, cyclin A, all of the cyclins playing the role. So in this case, embryo start transcribing its own RNA. This is a time, right? And cells start moving around in preparation for the gastrulation. So that is the phase that they will follow. So what determines this mid-blastula transition? The mid-blastula transition is hugely dependent on the nucleus to cytoplasm ratio or NC ratio. Now remember, at the beginning of the cleavage, that is the pre-cleavage time, where the zygote is formed and no cleavage is done. In that case, we have one nucleus, and let's say that zygote after 4 cell division will produce 16 cells. So that zygote we see the 16 cells that zygote will produce by transferring its cytoplasmic content. So here we have only one nucleus and capacity to produce 16 different cells. So if we see the value is 0.06, which is kind of close to 0. So the nucleus is to cytoplasm ratio is close to 0. But on the other hand, if you look at here in case of gastrulation, the cell division process, after 4 cell divisions, the number of nucleus becomes 16, as well as the number of cells that are generated also becomes 16. So the number comes like 1, close to 1. So this is the idea. The nucleus is to cytoplasmic ratio at the pre-cleavage condition was close to 0, but now after this process, after the mid-transition, mid-blastula condition, mid-blastula transition phase, it will become close to 1. So from 0 to close to 1. This is the ratio of nucleus to cytoplasm, which determines whether the mid-blastula transition will occur or not. So we have already talked about the embryonic stages of blastula like that, right? So now let's talk about another important fact. Say here, look at here, this point at the morula phase, where all those cells are stacked upon one another. And I've told you earlier also, those cells should interact properly with themselves, right? And the interaction here is cell-cell interaction. The cell surface receptors are present. With the help of those cell surface receptor, mostly proteoglycans, they interact with themselves. Now this cell-cell interaction is very important because at this point those cells should be placed together like a formation of a mass of cells or ball, because we don't want those cells to be separated from each other. So why those cells aggregated with each other? The reason behind that is a cell adhesion molecules, right, that influences the blastula organization. The cell adhesion molecules are also known as CAMs, right? These are transmembrane cell surface proteins that binds to the CAMs of another cell. So one cell have a kind of CAM, another cell, let's draw it with another color. So this is another cell, have another CAM. With the help of these CAMs, those two cells can interact with itself in the surface level, and that interaction is required. So CAMs are involved in cell migration as well as producing stable tissue structure. That is also very, very important. The experiments have shown that cadherins, cadherins is one type of CAM, and cadherins plays a very vital role for the stabilization of all those cells and interactions of all those cells. Like say one type of CAM, cadherin is one type of CAM which stabilizes the frog blastula we see. Now there are different types of cadherins that are found in the surface of cells. There are E cadherins, there are P cadherins, and many more types. Now in case of frog blastula, the E cadherin plays a vital role of interaction. A cadherin that is present outside the cell and interact with the nearby cell. This is the image. So say this is a control embryo where you see those cadherins are playing a vital role. Cadherins are okay, they are prepared, they are interacting with themselves, and the cells are tightly packed with each other together, creating this hollow sphere in middle. This is the blastocele, right? But here let's say by some mutation, if we take out all the cadherin out of the place, or all the this is the cadherin taking out condition where there is no cadherin present. So see, blastula formation halted and blastula formation is prevented, because that idea of spreading cells to the periphery, creating a pore in the middle and arranging all the cells tightly to control that region where the blastocele will be there, is lost when the cadherins are gone. So cadherins play a vital role, and in different experiments it is proven. For example, in this beautiful picture it is illustrated. Say sudden change in cell adhesion occurs in the 8-cell embryo, and in this condition we have an uncompacted structure of the embryo. So all of the cells start dividing, and once they divide, they are attached with one after another to form this kind of structure, right? But normally in blastula or gastrula phases, we see the structure like this. See, by looking at the structure, it's barely impossible to tell how many cells are present there, because all the cells are merged with one another, but their membranes are not merged though. Their membranes are separate and they are separate cells, but the interaction between the cells are so beautiful that they are interacting and forming a one unit kind of structure like that, more compact structure. So see, same 8 cell here as well as here in two different embryos, but see in this case it is more compact and more what we say position effective, because you know more space effective model, because in this case it occupies more space, but in this case it occupies less space. So that's another important thing. The space is a matter, and in that case many cells to be stored. So this is the way they do this. Another very good example here. These are all real life, real time pictures. They are not schematics. So see here, this is another case where we see embryos. Here the embryo with cadherins and all these things, right? Cadherins are present. So see, all the embryos kind of attached. By looking at here from the top view, we can say in this disease that the cells are, these are kind of one cell, like it looks like a one cell, right? As you can see, looks like one cell. We're looking at a giant cell, but actually in reality multiple cells are there, 8 cells are there. They are arranged one after another in this beautiful way of cadherin interaction. On the other hand, if you see here, this E cadherin plays a role in this particular case. So in the second case, what we do, we treat these embryos with antibodies that are blocking the E cadherin, because see, this is a cell and there are E cadherin receptors. So if I design an antibody that will block this E cadherin receptor, so in no way that this cell can interact with the nearby cell with cadherin. So as a result, blocking this cadherin with the antibodies, anti-cadherin antibodies, we will see structures like that, where we can easily distinguish the cells. You can see 8 cells can be easily distinguished in all those plates, and they are looking like the cells are separated. But in this case, all the cells are working together as a unit. So that's what a cell want, that's what an embryo want during the embryogenesis. So once we talk about all the stuff, we started with the 2n number of cells, which is a zygote. Remember, we start with a zygote. That zygote then have rapid cell division. After the rapid division, it produces what we call morula, the morula phase. Then that morula, those ball of cells start to arrange properly, and what they form is called as a blastula, where the cells are called blastomere, and it is a hollow sphere of cells, which is a spore in the middle, which is called blastocele, right? And this is blastula. So let's write it morula. So we begin from zygote, then we have our journey through morula to blastula. So from blastula, we've seen that nucleus is to cytoplasm ratio play a vital role in determining whether the blastula, those embryo will ultimately turn and itself into the gastrulation phase. So once the gastrulation phase is reached, then the blastula will modify itself. The gastrulation starts while the blastula slowly starts to have invagination of cells or furrowing, cleaving of cells, because all the cells that are forming this hollow sphere slowly start to invaginate inside. So whatever cell region that are present outside slowly start to invaginate inside, and that's called the gastrula. Actually, the idea of gastrulation is to provide three different layers of cells, ectoderm, mesoderm and endoderm, right? Ectoderm is the content of cell that presents at the outermost side, endoderm the content of cell that is present innermost side, and mesoderm is the cell layer that is present in between the ectoderm and endoderm section. So this is the beauty of gastrulation. While during the gastrulation, even before the gastrulation, the cells of blastula even know what kind of cell and organ they want to produce. So their fate are already determined there. So once their fate are determined, then those blastula cells will undergo this gastrulation phase, where the folding of cells occur. And this is important stuff where, up till this blastula phase, we see the cell division playing a vital role, as well as in the blastula we see cell-cell interaction using the surface receptor molecules or cadherin molecules play a vital role. But after the blastula, while we are talking about gastrulation, we will see the cell migration playing a vital role. Those cells will slowly start to migrate, right? Slowly start to migrate and creating folds in the blastula hollow sphere. As a result, it generates three different layers. And from each of the layer, different organs will start to generate. From ectoderm, it usually produces skin. It produces neural or nervous system. From mesoderm, it produces digestive system. From endoderm, it produces digestive system and respiratory system. From mesoderm, it produces muscle system as well as a blood vessel system. So see, all those different types of different layers produce different type of cells and organ system in our body. So see, we'll see different gastrulations. This is the gastrulation of sea urchin. See, this is the situation of blastula, right? So this is a blastula phase of cell, where you can see the peripheral cell and a blastocele in the middle. These are the blastomeres. And in any embryo, there are two different options. One is the animal hemisphere and vegetal hemisphere. Animal pole and vegetal pole. Now remember, the gastrulation means the folding of those blastula cells like a sheet and generating three different layers. The gastrulation phase always works from the vegetal pole. Gastrulation always works from the vegetal pole. Remember, for an embryogenesis, the cleavage always starts from the animal pole, where there is less yolk content. But gastrulation always starts from the vegetal pole. So here see, gastrulation, these vegetal pole cells are normal, in the normal condition. Now these cells slowly start to migrate towards the blastocele region. So as they slowly start to migrate, they start to create a pore here, that is called a blastopore, and then it's pushing itself inside. Invagination start to form inside, and as it is forming the invagination, that blastopore slowly start to increase its condition, and then they create three different layers properly after the movement, because see, the migration of cell occurs in from the both direction like that, and then they will meet with each other. So see, in middle we have now colored coded it with the yellow, because this is a completely different inner tissue now, and there is a pore outside, and in between there is a section of hollow chamber which is squeezed and forming a structure like this. Okay, so see, at this point this is the endoderm. This is the endoderm structure that is formed, and the blastopore remains there. The blastopore that is present here, this blastopore will ultimately produce either mouth or anus, depending upon the organism we are dealing with. And then that cavity that is generated due to the invagination of those cells, that cavity, this is the cavity, remember, in between this cavity is known as archenteron, and this archenteron will finally fold, and archenteron is will ultimately produce what we know as a primitive gut of that organism. And this is the ectoderm, outermost layer. The ectoderm provides us nervous system as well as skin. Endoderm, this is the yellow layer, it creates the linings of digestive and respiratory tracts, and this archenteron produces primitive gut. Mesoderm was not shown here, but actually this is the mesodermal region. This is the mesodermal region. So the mesodermal region, it produces the blood vessel, muscle, bone, liver and heart. So see, these are the embryonic stages for the gastrulation phase. It's not very difficult, though many students find it difficult, but actually it's very easy. It's simple. Imagine a hollow sphere of balls, and imagine those blastomeres, all those cells slowly start to migrate to create the cavity in between. This is the image again. So see, these are the blastopores start to form from here, cells start migrating, and they are creating the gastrula phase. During the gastrula phase, microtubule dynamics plays a vital role, because it's all about migration of cells, right? So in any cases, migration of cells require microfilaments, microtubules and actin filaments to work properly. So see, this is the ectoderm that is present, and once it slowly start to create the vagination, in that case all those cells slowly start to stretch itself, creating a larger, longer, thinner cells like that. And see, the microtubules, previously normal cells arranged haphazardly like any place like that, but after that, while the cells are destined to produce this invagination, those microtubules start to arrange itself in stretched long condition to create thinner and larger cells, longer cells, and those microtubules then slowly arrange itself. They will squeeze the cell up, and then they create this invagination. And this invagination you will see from the dorsal to the ventral view for the cells. And while all the nucleus present at one point, the ventral point of the cell, and the microtubules are creating the thin cell lines there. Now, if you look at the gastrulation of frog, it becomes much more clear, because in gastrulation frog, again, this is the blastula phase with the animal pole and vegetal pole. See, vegetal pole contains all those components, right? All the Yolk components there. And here at the middle of there are three different lineage of cells that are already being formed, because I've told you that before gastrulation, cells already get their code for the determination. That means what kind of cell they want to produce, what kind of organs they want to produce. So see here, the joint between this blue and this cell, there is a red colored cells, as well as there is a small pore in between from where the invagination start. We call it a dorsal lip of blastopore, the lip of blastopore. And this dorsal lip of blastopore, that joint of the dorsal lip of blastopore contains some stretchy cells called as bottle cells. Actually, the bottle cell guide the whole process of invagination process, and it's very easy to find the migration of cells by staining the bottle cells, because bottle cell migrates forward direction as the invagination slowly start to occur at this direction. So if we stain bottle cells, we can see the migration. So see, slowly these cells start to migrate, and the blastopore slowly start to engulf, and there is a cavity start to form. See, this is a small thinner cavity. It will become larger eventually. So this is the involution that start to occur, and slowly they start to form that archenteron or primitive gut. Remember the same thing. In case of sea urchin, we see the archenteron form straight forward. Here also we see with the help of a blastopore at the terminal, and this is the lip Dorsal lip means a portion which closes the terminal or closes the blastopore and opens the blastopore. It's a small tissue. So that is the dorsal lip. So here this archenteron that is formed due to the migration of cells will form the primitive gut again. But see, the migration of cells are in this direction. This is the arrow, right? Migration is in this direction. So here, but these cells that are present in the joint will migrate to the bottom, and these all cells will migrate to the top, which is a anti-clockwise direction in this case. But all the other cells that are present on the top, the ectodermal cells, that will migrate to the opposite direction. Okay, so see, the inner cells, they migrate, the inner tissue, that is the movement start, it will migrate to anti-clockwise, but the outer tissue, that is the blastomere cells, they will migrate to the opposite clockwise direction, right? As a result, what they will form, they will bring itself down, and this pore that was previously there, the blastocele content, which will be fused properly at the end, and that blank region, that small hole region that is there, which will be blocked by a clause called Yolk plug, right? And rest is only archenteron that is formed. Rest of this blastocelic portion will be sealed with the Yolk plug. Remember, this is a unique feature for frog gastrulation, which we haven't seen in case of sea urchin gastrulation, because in this case that blastocele section, as this part moving closer as well as this is moving closer, they will fuse with each other, and that blastocele portion is gone now. We only have whatever content remain, we only have with the archenteron, and that will form primitive gut. So now if you look at the gastrulation in chick or chicken embryo, what will process? Because chicken embryo division is quite different than that of the frog gastrulation, because in chick we know the blastula is not that much, form not that much, because the cell division that start to form in chick from the animal pole, if you see, they are incomplete cleavage that start to form, incomplete cleavage. So this is a cell from the top view. If you look, this is the animal pole top view. Cleavage occurs start like that, and after that second cleavage like that, So see, the cleavage is not complete in case of the chicken embryo. So the cleavage occurs in the middle and cleavage occurs sometimes. So this incomplete cleavage that occurs during the chick cleavage process or cell division process, and then it forms the morula, then blastula and all this stuff. So whatever thing is formed, it contains all these regions that are present. See, this is the structure, the cross section that is showing us the process. So see here, these are the blastocele, this is the blastomere cells that are present here, right? And here are the blastocele content, right? This is the blastocele content, and yolk is present there, right? And if you look at here, there are the already tissue that are being generated, which is the endoderm. This is the section of endoderm, and these are called the hypoblast and epiblast. Whatever cells present at the top, in case of chicken we call them epiblast, ep means in the top, and whatever cells present in the bottom region we call them hypoblast. So the idea here, again, in case of chicken, the process of gastrulation is related with the development of neurons. The neural development is tallied with the gastrulation phase, where the neural node that is formed due to the cleavage, the neural node forms. See, this is the neural node, which we call as a primitive streak. The primitive streak means small fold originates on the top of the embryo, and that from that fold, see, the arrows that are denoted are the migration direction for the cells. So all the surrounding cells, they migrate to the bottom like this, the bottom region like that. So if this is the surface, just imagine this is the surface, and this cells slowly start to invaginate, create this invagination, and slowly start to go to the bottom like that. So what it creates a V-shape like structures, the invagination there. That's what's formed here in the primitive streak, right? So see the cells migrating in this direction, and all the cells are migrating towards, some of the cells are migrating towards endoderm, some of them migrating towards the periphery. So some of the cells which are migrating towards the bottom will contain and attach to the endoderm cells and convert itself into the endodermal cells. Now if you look at the development of neurons and the nerve system, which is very important, because in case of mammals as well as chicken and all these cases, the development of neurons are playing a vital role. The development of nerve system plays a vital role, and it is related directly with the gastrulation phase. So let's check it out. So this is the neurulation. The process of nerve development or neural system development is called neurulation, right? And this is the neurulation in frog. So if we see, this is the process of gastrulation again, but at the beginning there are some neural plate start to form. This neural plate that is formed, which will be the forebrain, the brain section of the frog, that will contain the cells which will ultimately destined to produce brain for the frog. So see, this is the neural plate of the brain, and this neural plate, see, it's just on the top of mesodermal cells there. These are the ectoderm, endoderm, these are the mesodermal cell, remember, right? And here this is the mesodermal. The red colored cells denoted here is the mesodermal, the blue colored cells ectodermal, and the green cells, this is the endodermal cell, right? So here this is the neural plate originated. So once the neural plate originates, see, the neural groove originates there. The neural plate formation is just like a formation, again another invagination. In any cases, it's a part outside now, like that. So what it forms actually, it slowly, so say this is a membrane, this is the cell membrane, and this is the invagination, so slowly downward section like that, it's the neural plate. Then cells start to grow surrounding that neural plate. These are the neural plate lies in between. Cells start to grow surrounding it, and then slowly join itself, engulfing the neural plate, creating a hole like structure in between. So see here, this side and this side, from both these sides cells start to grow and migrate, and then join to finally embed that neural plate in the middle, inside, right? So see here, this is the blastopore, means this is the section of blastopore. If you look at the top section, and this is the neural groove that is formed. Neural groove means, again I've told you, the formation of neural groove is due to the increment or increasing cell content from surroundings, while the neural streak that is present in the middle, and the cells start to migrate, and it is engulfing the streak. Simply something like that. Say this is it, and the neural streak, or let's say this is the neural plate, whatever, and then cells start to, so in the middle, and cells start to increase in both these cases, like this. See, slowly start to rise itself up. And then very slowly, and ultimately it will fuse itself. So see, if I tell you this is a process, schematics, it starts with that, this is the neural plate, and slowly the cells slowly start to rise from both this terminal, and this rising of cells, again it's important for the function of actin filaments and microtubules. They start arranging the cells, pushing the cell membrane, pushing the cytosol into one direction, and cleaving the cells in the top. So once the cells start to rise from both the terminals, start to rise, and then slowly it gets itself closer and closer, and then fuse. These two cell sections will fuse, holding this is the region where the neural plate is inside a protective vessel, like that. So here this neural plate will develop into brain, and this process does not work in a particular region. I mean, it's not work only a point of the embryo, it occurs throughout a stretch of the embryo. See, this is the embryo. This whole process of neural plate is placed through the top in a region, and all the cells that are arised from both the sides will be from the two sides of the cells. So just imagine this whole process working. We are looking at a cross section in this particular case. So just imagine throughout the embryo we have this neural plate and neural streak going on, primitive streak going on, and those cells start to arise and they'll fuse to hold that thing in a chamber, to hold that whole neural streak in a chamber, inside a chamber, and this is the chamber, right? So it will form a structure with a chamber like this. Inside that chamber we have that neural plate and all the neural systems. That thing will in future will develop into, that neural plate will develop into brain, and the rest of the thing will develop into the other neural system, the central nervous system content. It will develop into the central nervous system, and other cellular structures will give them the rigidity, just like our backside. If you just imagine in case of ours, in the dorsal side of ours, right, we have the same thing. This is how the neural condition generates, because again in case of frog, see, the engulfment is done properly, because see in this case, this is the primitive streak region and these are the tissue layers that are present. See, this whitish tissue layer slowly start to go inside, folding is occurring while these green structures are coming out, the green layers are coming out, up, and slowly it's engulfing that neural streak along with the white region, and it's engulfed it completely, it's joined itself completely, it's fused of the neural. So neural folds slowly start to arise, neural fold and arise, and neural groups start to form, and then finally neural folds are joined, creating a tunnel inside which the neural cells are present, right? That's how the structure generates. This is the process of neurulation. See, this is a process, it's also depicted here with this picture. See, if I erase all this, or you can see it also. These are the different layers, ectoderm, endoderm, and see, this is the notochord. Notochord is a section which is then modified, from where the modifications start to occur. Say, these are the neural folds start to arise. The neural fold, see, the neural fold cells are nothing but the ectodermal cells. So that's why I told the neural content, the neural system for an organism developed from the ectoderm. So see, the ectodermal neural folds start to form, then slowly start to rise, the neural, they slowly start to rise, and the neural groups start to form, and then those membranes will fuse with each other. See here, they are fused with each other, and we see the formation of neural tube. This is called a neural tube. While the neural tube is formed, inside the neural tube we have the neural plate and all the neural system, right? So see, during the development of neural groove and neural fold, right, or ultimately what we call as a neural tube, the formation of neural tube, it's very important that cytoskeletons play a vital role. Because see, in the normal condition, cytoskeleton remains like this. These are the scattered condition of cytoskeleton. But then they slowly start to organize. The microtubules organizes itself and stretch the cell so that the cells start to arise from both the sides. See, the cells start to arise, then slowly start to migrate. The cells start to migrate, and then finally they will be fused, creating a new neural tube. See, this is how the neural tube forms. And those cells that forms the neural tube, right, the neural cells, they are the ectodermal cells. This is a tissue section of the frog neural generation, for the neurula phase actually. It's also called as neurula because the neural development is going on. The exact image that we saw earlier, so I'm not going to talk about that here. See, this is the neurulation in cheek embryo. We have seen the neurulation in frog. Now in this case, the neurulation in cheek embryo. The process again remains the same kind of process, right? So say this is the gastrulation phase where the migrating cells are there and primitive streaks start to form. Remember, I've talked about primitive streak earlier. The cells are folding in the bottom. They're passing and migrating to the bottom while creating this invagination in the bottom. So during this formation of these structures, so see here, all these cell layers, the endoderm, the yellow colored endoderm, the red colored mesoderm, as well as the blue colored ectoderm, all the cells start to arise. They start to arise. See, endodermal and ectodermal cells, endodermal and mesodermal cells slowly start to arise, and while they start to go arise their content, because the formation of neurons and the neural tube in case of chick embryo is slightly different, because in case of frog and all this case, what we've seen, the process occurs after the formation of ectoderm, and then ectoderm slowly start to rise and fuse. But in case of, so it's the rise and fuse, this is the formation of neural tube, right? So normal content, then slowly they start to rise, the cells start to rise from the side place and then slowly engulf it, creating the neural tube. While in the other hand, in case of cheek gastrulation phase, the cheek gastrulation works in a different way. The invagination takes place in the bottom like this. It's not like up, it's go down like this. So while it's going down like this, during the process, this primitive streak region of both the ends start to fuse. Once they fuse with themselves, it is creating the neural tube. So in this case, how this process works, again let me draw. So it works like this. These are the cell lines, and this is the arrow, the direction of the migration of the cells. So while the cells migrating like this, and then finally like this. So see, if this is a neural cell that is present in the middle throughout, see, this is a process where the red region is the neural plate that we are talking about, and the outside, these are the ectodermal regions. So this is the ectoderm, and this red region, neural plate. So see, neural plate is in the middle somewhere there, and the cells are migrating in this bottom direction. See, this is the direction of migrating. Cells are migrating, they keep on migrating and keep on very close to each other, because the cells, so they drag the cell from surface, they keep them very close to each other, and after some time when those two cells, those membranes fuse, the ectodermal cell layers fused with each other, then it forms a cavity containing the neural fold inside. That cavity is called the neural tube. That's how the neural tube is formed in this case. Remember, for the formation also, they contains the bottom layer also slowly start to rise during this process. See, in case of frog, we see already the cells start to rise from both the sides, but here also the cells start to rise, but it forms the groove like structure. So if you just see the top region of the embryo of chicken, so it will look something plain like that, and slowly you will see something to rise, the structure like that. That structure through the top of the embryo, if we design the embryo from the top, if you see the embryo from the top, you will see a rosed structure from both the regions. It's a raised structure. See, this one, it's a raised structure, and this structure will form, and they push lot of cytoplasmic content, lot of Yolk content to the terminal to push the cells to rise, and this is called the Yolk stalk. You can see the Yolk stalk where it forms a stalk like structures for the structure to hold that structure together. It pushes a lot of cytosol there. So this is how the process of organogenesis start to occur, and the neural tube start to form in case of chicken. And that neural tube that formed, see, this is the neural tube. Again, the neural tube is a large neural tube from the top to the bottom. So see, that neural tube will contain some section of neural plate cells and the rest of the other regions. So the neural plate will turn into a brain and the optic system, optic vesicle and all these things, and then rest of the part will slowly turn in themselves into the spinal cord region, which contains all the important neural developed conditions in there. So that's how the spinal cord is also formed. So we have talked about all this process from a zygote or 2n number of chromosome containing cell, rapid cell division, morula, from that blastula, arrangement of cells creating a hollow sphere of cells, blastula. Then it converts itself into the gastrula phase. Gastrulation is a process which produces or introduces us to ectoderm, endoderm and mesoderm, then finally differentiates cells into produce different organs. So the determination, remember we already talked about determination in very first section of the video. Determination means while the cells are destined to produce a specific organs or to produce into a specific mature structure, once it is destined there, that is the determination, and determination is required for the differentiation. Determination is required for the differentiation of cells. Once the differentiation of cells are very important, once the differentiation of cell is done properly, then they are matured and they turn into the gastrulation phase. They turn through the gastrulation phase, and then only the morphogenesis takes place. Morphogenesis means once the cell destined to produce a specific type of mature organ or structure, then there will be some changes over time, some chemical changes, as well as the genetic interactions will go on, new gene products will produce, genetic expression will vary, and it will produce that particular organ that they destined to produce. That's about the morphogenesis. So see, morphogenesis is extensively studied in drosophila. So see, morphogenesis contains pattern formation, while the organization of differentiated tissues, so that's what I talked about. Morphogenesis occurs after the gastrulation phase, while all the tissues are already differentiated, and then they'll produce the specific structures. For the body segmentation in fruit flies, right, the gradients of materials established in the egg and the actions of series of embryonic genes that place there to finally turn themselves into the specific structure. For example, in the fruit fly, there are different segmentation of the body. You will see in case of insects, multiple segments of body are there, like head, thorax, and the abdomen. So these are the three major segments. Even in each of those segments, like in the thorax, there are mediothorax, ultra bithorax, bithorax. So different thorax regions are there, mid thorax or the top thorax and the bottom thorax, different regions. And in abdomen itself, if you just study the abdomen of an insect, you will see, or in case of a butterfly, you will see a structure like this. That abdomen is further divided into different fragments. So how come they produce lot of different fragments? And not only they produce that organ, but also that organ has a specific way of handling and dealing with the situation, because the abdomen will never have any of the legs. Leg always come from the thoracic region, not from the abdomen region. So these things are required, and how could they arrange this thing? This arrangement completely depends on the gene expression process. The gene expression, there are multiple genes that are working together, they're interacting, the gene products are interacting together to finally produce and produce a segmented condition, the segmentized options for the insect body. So say at the very beginning, and also as the insect larva is a centralized yolk content, it also needs to determine which part will be the axis, anterior, which part will be posterior, which part will be ventral, and which part will be dorsal. That's very important, right? So see, in this case, if this is the organization, that body axis determination also is an important task in case of this fruit flies. So we have a different video about fruit flies completely dedicated to them, where I've talked about all this process like body axis determination, as well as the differentiation, as well as the body segment patterning. But in this video, I'll majorly talk about how the segmentation works in a basic level. So there are genes called the maternal rule genes or maternal effect genes. Then maternal effect genes, and there are some genes called gap. This is an example of such gene. This gap gene influence some other types of genes, which will add some basic crude concentration gradient of proteins. For example, here you see the concentration gradient of this red color. Here you see the concentration gradient of this blue color proteins. And at the middle, none of the proteins are present, designating it at the middle center, and the red present more in the anterior, blue present more in the posterior. So this type of patterning or the gradients start to form by the gap genes. Then a gene came pair rule genes. Pair rule genes then slowly segmentize the body into different regions, and then the segment polarity genes comes in. So if we begin with that, maternal effect genes establishes the gradient in cell. Then gap genes define the broad areas that will be regulated by other genes called pair rule genes. So see, this is a function of maternal effect genes, like the example is becoid. Becoid is a gene product which is present in the anterior, designating anterior. Nanos is another gene. Oscar is another gene that is determining the posterior. Now the gap gene comes in, it segmentized a portion. It defines an area or the region which will produce the different segments of the body. Once that is designated, then other type of genes come in which are called pair rule genes. Now the pair rule genes here, they refine the segment locations, small segment location, which part is going to produce what. Thus all segmented locations of the body are made there and regulate the segment polarity, and then it brings the other gene called the segment polarity genes. Finally, the segment polarity genes along with the homeotic genes, see, these are the two genes that work at the downstream level. Segment polarity genes and the homeotic genes work together to determine which will be the, among them the segment polarity genes determine the boundaries of each segment and homeotic genes define the role of each segment. So once the segment polarity gene determines that this is the boundary, this see, this is one of the lining, this blue line. So this is the boundary for that gene, and homeotic genes determine that this is the section is going to be a part of the thorax and it will be the portion where the wings will be attached, and then they'll bring it to that kind of shape as this drosophila goes by, as the morphogenesis goes by. So it begins with the maternal effect genes. Maternal effect genes at the top controlling gap genes. Gap genes are controlling pair rule genes. Pair rule genes are controlling segment polarity genes, and segment polarity genes are controlling homeotic genes. And segment polarity, homeotic, both of them are controlled by pair rule genes. They finally brought about the changes in the segmentation. Remember, it's not like that, that this is the part, because so if you have a particular, say I give you a cake and I want you to cut into specific locations, specific regions, and design it accordingly, you need to have specific instructions, like which part will be what, where you exactly cut it, which will be the boundary of it, which will be the size of it, and then where you exactly cut and which place you design in which way. So those are the instructions. See where exactly you will cut, which will be the side, which is, so see, this is a straight large cake. If I give you a straight large cake like that, like this kind of rectangle cake, you should not cut it vertically, horizontally like that. You should cut it like this, right? This is the idea. This is provided by maternal effect genes there at the beginning. These are the terminal, this is anterior, posterior, so you should to cut that way. Then the gap genes will come in, which will tell you where to cut it, right, in the different area. So if you want to cut in small segment like 20, if I order you to cut into 20 different segments, same length segments, in that case, for a better understanding, what will you do, you cut it from the middle first, then you cut it from the half again and half again to get the exact same content in each egg, in each cake actually. So for that reason, you will first break them down into broad areas. That is done by the pair rule genes. Once the pair rule gene functionality is done, then what you will do, one of the broad area you take. So see, from this cake you will develop say 5 you'll develop 5 same segments, 5 same segments you develop, and take each of the segment, then you bring the pair rule genes there. Pair rule genes define the segment locations, right? So you take each of this peg, and segment polarity genes help in determining the boundaries. So now you know you have to cleave each of them into 4 pieces. So you'll start cleaving them into 4 pieces, see, like this way. And then homeotic genes means defining the role of the places. So once you cleave them based on the segment polarity gene sections like that, you cut them into same pieces. Then the homeotic genes, what will they do? It will define which section will contain and which will be the role of each segment. So now once you cleave them, among 20, you decorate 5 with cherry, you decorate 5 with raisins, decorate 5 with cashew, decorate 5 with walnuts. So that's the way each of the segments will be decorated, their functions will be defined. So nothing, no analogy can be more convenient than the cutting a cake here, because this is the way you can understand how the gene actually functions, right? I see many students confusing all the terms, maternal effect genes, gap genes, pair rule genes, segment polarity genes, and homeotic genes. This is how these genes actually work, right? Okay. Mutation in a homeotic genes cause lots of problems. So if we have a homeotic gene problem, even in some cases we see legs coming out of the head and all this stuff. There are some mutations. I'm not going to talk about the details here about that, but mutations in homeotic genes can create problem with the function. Though the genes are defined and determined about their function, but if we place them in the wrong regions, wrong mutations, in that case they will produce a different function. In head region, the homeotic genes that should provide properly in the head region, it will develop into the different portions like antenna, right? But in this case, due to the mutation, sometimes they will generate leg instead of antennae. In intrinsic homology found between the genes controlling the segmentation of body. And if you take mouse embryo as well as the fruit fly embryo, in both this case, if you see the 10-hour and 12 days embryo for both of them, you will see some extensively region. That this is a green region which will make the head portion. This orange region which will make the back region, like that. So see, this is the genetic content. This is the fly chromosome. This is the mouse chromosome. You see a lot of similarities out there. This is the homology that is found. It is found that those homological portions, those genes, are kind of fixed from the insects. You know, insect and mammals are distantly related, but still you see this is homology that is present in insect as well as in mammals. So we found that those genes are very, very important for the development as well as for the development of organs and the proper functioning, right? And in the morphogenesis process, it requires the apoptosis to work properly, because during the morphogenesis we not only need, sometimes we need to generate some cells, right, and sometimes we need to kill some cell for the process, because it's all about trimming. Let's say you have a pile of, say if you have a play dough, I mean if you have dough that is clay, Chinese clay. Using Chinese clay we design different things, like excuse me. So if you have the Chinese clay and if you want to design different things, sometimes we need to add some Chinese clay, sometimes also we need to create a curvature, we need to cut it and also exclude some amount. So we need to add some cells or we need to delete some cells also during the development. For the deletion of cells during morphogenesis, we rely on programmed cell death or apoptosis. Where you say in case of the development for frog as well as for the duck, most of the cases. In case of duck, the process, say in all case of duck as well as in case of human, in both the case, if you see the development, during the development, all of us have this kind of structure, the joint structure, because cells start dividing and making the structure. The individual fingers are not properly made, but as the time goes by, say after 41 days of fertilization, you have limb structure similar to that of duck. But in case of duck, that structure remains as it is, and that structure slowly start to grow into the adult duck. But in case of humans, after 56 days, in between that 15-day duration, all those joint regions of the membrane slowly start to undergo apoptosis or programmed cell death. So those cells start to die, leaving only this small region of cells which we call as fingers. Okay. So finally, this is a conclusion time. The conclusion about the cellular and molecular mechanism that is essential to development is the specificity of receptors and enzymes that are present. From the beginning of fertilization, we need receptors to be present on the surface of our gametes, like receptors should be present on the surface of either egg or sperm for proper fusion. And all we need enzymes in form of digestive enzymes present in acrosome for mediation and delivery of the sperm pronucleus inside the egg. Then cytoplasmic organization is a beautiful thing. It is a very important thing, because otherwise a proper cytoplasm organization, cleavage will not take place. For a proper cleavage, we need to form two sections where we have a yolk rich region in one hemisphere and yolk less region in the other hemisphere. And the cleavage will start from the animal pole where we have a less yolk. Cell movement through the action of cytoskeleton elements and cell adhesion molecules play also vital role. Cell adhesion molecules play the vital role for the compactness of the cells together in the morula as well as in the blastulation phases, so that the cells will not segregate, so that the blastocele content maintained as it is, as well as the cytoskeletal like the actins as well as the intermediate filaments and the microfilaments play the vital role, because the microtubules not only help in segregating the chromosome during the rapid cell division, but also those microtubules help in pushing the cells, the side cells, the ectoderm forward, so that to produce the neural tube or the neural development during the neural development, as well as it plays a vital role in the cleavage and farrowing, as well as it plays a vital role during the invagination of the surround blastomere cells to produce the gastrulation phase. And the orientation of tissue layers leading to the induction. Induction means one particular tissue influence another tissue to grow into a specific organ. That induction, we've seen that the forehead of the frog embryo influences the nearby tissue to grow into the lens, right, the optical system for the frog. And in this case, the orientation of that layer is very important, because that layer pushes the other tissue to form a specific structure. So see, this is the interactions in developmental biology. The interactions is based on the receptors, the enzymes, cell-cell interaction, genetic interaction. We see more in case of the development of drosophila, in case of the segmentation of the body, in case of the organogenesis process. And all of these things work together to finally give us a healthy, mature individual. And then they'll start to grow again after a time until it's adulthood. So gene expression at the end, and that's how we complete the whole process of developmental biology basics. And I think if you learn all the stages, all the different part of the video that I've covered throughout this process from the beginning till this end, 57 slides are there. I think if you study it properly, the way I told you, I think you'll understand developmental biology. If you're a microbiologist or biotechnologist, probably you don't know much about developmental biology before, but this will give you a clear-cut insight of developmental biology. So if you like my videos, please don't forget to subscribe to my channel, like the videos, and share these videos with your friend so they can also get this idea. Thank you.